right, we are continuing chapter 13, and we are going to cover hearing, balance, and vision in this video. So a lot of this will also include some of the lab terms that you needed to learn recently in lab, but this will be kind of a nice supplement to that. All right, so first up is the ear, so the sense of hearing, and this is one of our special senses. So a lot of this initial stuff was covered in lab. So we have that external ear with the auricle, the auditory canal, and the tympanic membrane. So the auricle, that external part of our ear, is really important for helping to move sound waves into the auditory canal, which will move them to the tympanic membrane, which is going to pick up those vibrations, those sound waves, and transfers or transforms them into mechanical waves, which goes through the middle ear, which is the space spanned by the three small bones called the ossicles. This was the malleus, incus, and stapes. These are conducting those mechanical waves from the tympanic membrane to the inner ear for the beginning of the processing step. The middle ear is connected into the pharynx through the eustachian or auditory tube, and this helps to keep air pressure equal across the tympanic membrane, so both on the external side of the membrane and the internal side of it. And so when we have a lot of pressure or fluid buildup in this eustachian tube, this is what's going to cause that plugged ear sensation, because it's not able to help regulate that air pressure difference. The inner ear is a bony labyrinth with some membrane sections in it, separating out tubes and spaces. Space in between the bony and membranous labyrinth is filled with a fluid called perilymph, and so this is very, very similar to the lymph that we would find in the rest of our body, but it has some slight differences in its function here, obviously. So this is really similar again to that extracellular fluid, but we do need some of this fluid in the ear. And then inside of that membranous labyrinth is going to have endolymph. So inside of it, and it actually has a really, really high level of potassium in it, but very, very low sodium compared to the perilymph and the rest of the extracellular fluid. And that makes it more conducive to moving nerve response or impulses around. So it's better having that differences in potassium and sodium concentration, makes it easier to move those nerve impulses around. So the inner ear has three regions: the cochlea, vestibule, and semicircular canals. So the cochlea is the section responsible for our sense of hearing. Then the vestibule and semicircular canals are both responsible for our balance. But the inner ear is ultimately responsible for transforming those mechanical waves picked up from the tympanic membrane into electrical signals and getting them sent to the brain through the vestibulocochlear nerve, or cranial nerve number 8. So here is our graphic. External ear with the auricle, ear canal, tympanic membrane. The middle ear has the malleus, incus, and stapes, and then the eustachian tube. And then here is our inner ear with the cochlea, that little shell shape. The vestibule here at the base of those semicircular canals. And then here is the nerve. So we do have two initial separate nerves, the vestibular nerve and the cochlear nerve, and then those do end up fusing together to move the rest of the way to the brain. So actual hearing, or audition, is what this is referred to as. Again, this is the job of the cochlea. So it's that spiral-shaped tube. It has three compartments to it: the scala or scala vestibuli, scala tympani, and cochlear duct. And all of those compartments are filled with that endolymph. So it's going to have that really high level of potassium, really low level of sodium. And so it's going to help with transferring those nerve impulses. So the stapes, that little bone, is attached to something called the oval window, which is located at the very beginning of the scala vestibuli, which will then extend above the cochlear duct. And we'll see a graphic of this here in just a moment on the next slide. And then at the most upper tip of the cochlea, that vestibule curves over the top of the cochlear duct and becomes the scala tympani, and that will return all the way to the base of the cochlea, traveling underneath the cochlear duct. And then that scala tympani ends at something called the round window. So as the vibrations, those mechanical vibrations from the ossicles, travel through this structure, it's going to pass through the oval window first, and then it will move through that fluid, that endolymph in the scala vestibuli and tympani, in a wave-like motion. Then it's going to reach that cochlear duct containing several organs of Corti, are what they're called. So these are going to do something called transduction. So this is basically kind of converting that wave-like motion of the fluid into the actual neural signals, those nerve impulses. So the organs of Corti lie on top of something called the basilar membrane. This is the side of the cochlear duct located in between the organs of Corti and the scala tympani. And so it is going to just help separate those two structures apart from each other. That's usually what membranes do. And then the organs of Corti are going to contain specialized hair cells. So these are named for the hair-like stereocilia that extend from the cell's top or apical surface. So stereo, sound. So these are special cilia that are responsible for some of this hearing sensation. So they're going to extend up to a structure called the tectorial membrane. So this is going to be attached to the medial side of the organ of Corti, and they are still going to kind of do the same job, is helping to transfer that sound up to that second membrane. So when those pressure waves from the fluid move the basilar membrane, so they'll kind of push up on that membrane a little bit, the tectorial membrane is going to slide across those stereocilia, bending them either towards or away from other structures, other cilia, and this is what's causing electrical signals to get generated in that cochlea. So here's a cross section of that cochlea. So within these tubes are all of these different structures. So this kind of bony section out here, that is the cochlea. And so you can see here we have the scala tympani. Here's that basilar membrane. Here's the tectorial membrane right here. Organ of Corti in between them. So here's those cilia right there. So as this basilar membrane, as that fluid kind of pushes it up, it's forcing all of these cells and those cilia up and moving that tectorial membrane to get those electrical impulses. And then right here's the cochlear duct in the middle, and then the scala vestibuli up here. So these are all going to be full of that endolymph again, which is having those wave-like movements as it passes through these different areas. So here's some histology. So this is a stained image under the microscope of real tissue out of a cochlea. All right, so here is the vestibular membrane, here's the tectorial membrane, here's that organ of Corti again, so we can't see the cilia in here, but they would be up here, and then that basilar membrane right there. So here's a zoomed in view on those hair cells in the organ of Corti. So here's those hair cells right there. And then we've got those stereocilia sticking up. tethered to that tectorial membrane. And so again, that's all going to help generate those electrical signals to actually get that sense of sound, because we do need to convert all of those sound waves into electrical signals that can be sent to the brain for processing, because our nervous system runs again on electrical signals primarily. We do have obviously a few chemical signals, but it's mostly electrical signals. So here is that transmission of sound waves, so kind of a graphic showing how this process works. So first step is we have some sort of sound that we hear. So the sound wave shown here is representing alternating areas of high and low pressure. So that is going to basically, the high and low pressure areas, how often they occur is going to determine what we're hearing, how loud it is, and all that kind of stuff. So all of those sound waves pass through that external canal to the tympanic membrane, causing vibrations. So those sound waves will cause that to vibrate. It will transfer those vibrations across the ossicles, and the ossicles are also going to help amplify those vibrations, so making them much stronger. And then they will vibrate against that oval window, which is going to start causing that fluid, all of that lymph fluid, to start moving in more of a wave-like fashion. So here is that first region, the scala vestibuli, cochlear duct in the middle, and then the scala tympani section on the bottom. Here's that basilar membrane, organ of Corti right there in the cochlear duct. So the frequency of these waves in here are going to be the same as the sound waves were initially. So as that pressure bends that membrane of the cochlear duct, that is going to cause those hair cells to vibrate. And then that will transfer everything into electrical impulses, which can then be passed through the vestibulocochlear nerve and into the brain. All right, next up is equilibrium or balance. The vestibule is the first part of that inner ear that is responsible for that. So balance is another of our special senses. So again, we have hair cells with stereocilia on them functioning as mechanoreceptors here. The hair cells are located in a special structure of that vestibule called the maculae. So we have the urticle and sacculae sections of that. So these stereocilia here in this region are going to extend up into a very viscous gel, so kind of a very thick gel-like substance called the otolithic membrane. This is on top of a layer, this is a layer of calcium carbonate crystals called otoliths. So the otoliths make this membrane very, very top heavy. And so that is going to allow it to help move separately from this macula, which is going to help with some of those sound movements, or not sound movements, head movements, sorry. And so this is going to help keep our balance. We'll see some graphics here in a little bit. I know it's kind of hard to visualize what's going on without some graphics and just reading text. So the vestibule is going to sense what's called linear acceleration. So basically movement of the head in some sort of straight line, one direction or another, as well as gravity. So when the head moves, those otoliths move, and then the otolithic membrane is going to bend those cilia on the hair cells. So here's our graphic right here. So we're looking at this region. All right, there's the macula. So here's a zoomed in view of that top of the macula. So again, we have endolymph in here. Here's those cells of the macula. We've got hair cells down here at the bottom, going up into that otolith with some calcium carbonate crystals on top of it. And so if the head's upright, here's those hair cells again with that otolith and all of the calcium crystals up there. So the head's upright, these are all sticking straight up as you can see, and then when you tilt the head forward, then that force of gravity is going to put pressure on those cells, on that otolith, which will transfer to those cilia, and those will transfer down to the rest of the cell and out the nerves down here. All right, so this, again, mechanical movement is getting translated, transduced into electrical impulses. And then the semicircular canals are also really, really important for our sense of balance as well. So there are more hair-like cells located in the semicircular canals. In the ampullae, this is at the base of the 3 semicircular canals. One of these are oriented in the horizontal plane, the other 2 are oriented in the vertical plane. And the stereocilia on these hair cells extend into what's called the cupula or cupula. This is a membrane attached to the top of that ampulla. And these are going to do more sensing head rotation, so turning side to side, nodding up and down, rather than that linear movement. So as the head rotates, that's in a plane parallel to the semicircular canal, the fluid's going to lag. So the fluid's not going to move as quickly because we're basically moving in the same plane as that semicircular canal. So it's going to deflect the cupula in the opposite direction of the head movement. So if you turn your head to the right, that cupula would move to the left. And so it'll bend the stereocilia also to the left. So right here, here's that ampullary nerve. So it's going to help pick up all of these signals. Here are the cells. Here's the cupula. So in the still position, all of that's stationary. And then as the head rotates in one direction, so you can see if the head is rotating here, this would be to the left. This is a mirror image. So this would be head rotation to the left. And so you can see that rotation, the cupula is going to bend in the opposite direction of the rotation. So in this case, I know this graphic doesn't make it the most obvious, but this would be going to the right. So we are not looking at a mirror image here, but we are up here. And then that movement, again, gets transferred into electrical impulses to be sent into the brain. All right, our last topic for this video is going to be vision. Actually, you know what? We're going to call this video here and continue vision in the next video.